video, we are going to learn about cell cycle and cancer. The act of cell division is the culmination of a series of events. The period between two mitotic divisions defines the somatic cell cycle. The time from the end of one mitosis to the start of the next one is called interphase. The period of actual division corresponding to the divisible mitosis is called M phase. The interphase is divided into the G1, S, and G2 periods. Cells may withdraw from the cell cycle into G0 or re-enter from it. The timing of the four stages varies from species to species, from organ to organ within a species. In most cells, the whole of M phase takes only about an hour, which is only a small fraction of the total cycle time. The interphase is divided into G1, S, and G2 periods. Cells may withdraw from the cycle into G0 or re-enter from it. The timing of the four stages varies from species to species, from organ to organ within a species. In most cells, the whole of M phase takes only about an hour, which is only a small fraction of the total cycle time. Then we could see a picture depicting this. The four successive phases of a standard eukaryotic cell cycle. During interphase, the cell grows continuously, and during M phase, it divides. DNA replication is confined to the part of interphase known as S phase. G1 phase is the gap between M phase and S phase. G2 is the gap between S phase and M phase. Cells in G1, if they have not yet committed themselves to DNA replication, can pause in their progress all around the cycle and enter a specialized resting state, often called G0, where they can remain for days, weeks, or even years before resuming proliferation, or quiescent state. Here we could see a picture, eukaryotic cell cycle, which just represented in a circle. So G1 phase is there, gap 1, cells that cease division, G0. Then S phase, DNA synthesis happening there. Then G2, gap 2 phase, then mitosis. So mitosis G1 S G2, mitosis G1 S G2. That's how this cycle just goes on and on. And after that, cell cycle. Cell cycle is an ordered series of events. There are four main parts of the cell cycle: M phase, mitosis, and three parts that are components of interphase: G1 and the gap period between the end of mitosis and the start of DNA replication, S, the period during which DNA synthesis occurs, and G2, the gap period following DNA replication and preceding the initiation of the mitotic prophase. The engines that drive progression from one step to the cell cycle to the next are a series of protein complexes composed of two subunits, a cyclin and a cyclin-dependent protein kinase, abbreviated CDK. Cyclins are so called because they undergo a cycle of synthesis and degradation in each division cycle of the cell. In every eukaryote, there is a family of structurally and functionally related cyclin proteins. Cyclins bind to CDK molecules and control their ability to phosphorylate appropriate target proteins. There are three major classes of cyclins, each defined by the stage of cell cycle at which they bind CDKs and function: G1 cyclins, S cyclins, and M cyclins. Cyclin-dependent protein kinases also constitute a family of structurally and functionally related proteins. Kinases are enzymes that add phosphate groups to target substrates. For protein kinases such as CDKs, the substrates are proteins. CDKs are so named because of their activities are regulated by cyclins and because they catalyze the phosphorylation of specific serine and threonine residues of specific target proteins. The target proteins for CDK phosphorylation are determined by the associated cyclin. Because different cyclins are present at different phases of the cell cycle, so different phases of cell cycle are characterized by the phosphorylation of different target proteins. The phosphorylation events are transient and reversible. When the cyclin CDK complex disappears, the target proteins for CDK phosphorylation are determined by the associated cyclin. Because different cyclins are present at different phases of the cell cycle, so different phases of cell cycle are characterized by the phosphorylation of different target proteins. The phosphorylation events are transient and reversible. When the cyclin CDK complex disappears, the phosphorylated substrate proteins are rapidly dephosphorylated by protein phosphatases. There are three major classes of cyclin CDK complexes that control passage through the cell cycle: the G1S phase and mitotic cyclin CDK complexes. In yeast cells, a single CDK protein binds all classes of cyclins and drives all cell cycle events by changing cyclin partners at different stages of the cycle. In invertebrate cells, by contrast, there are more than one CDK to interact with G1 cyclins, S cyclins, and M cyclins. Then examples of selected cyclins and cyclin-dependent kinases: CD like CDC13, G1 cyclin, mitotic cyclins, S-phase cyclins, G1 cyclin, late G1, and S cyclin like CDK4, CDK6, CDK2, CDK1, mitotic cyclin, cyclin B. When cells are stimulated to replicate, G1 cyclin CDK complexes are exposed first. This prepare the cell for the S phase by inducing enzymes synthesis required for DNA replication and S phase cyclins and CDKs.